The last video for this lecture is a series of drawings that walk us through the process of translation. In this first drawing, you can see how the artist has depicted the ribosome. It's in these two big red molecules. Here's the small subunit, and here's the large subunit. They're showing us a piece of mRNA in green, and they're showing us a tRNA right here, sort of folded up like a clover, a three-leafed clover. You can see at one end of the tRNA, an amino acid is coming in to bind, methionine, and at the other end, there are three bases. In this case, UAC. Remember, the three bases that you find on a tRNA are called the anticodon. So this is the tRNA that carries methionine, and it has an anticodon of UAC. Notice that there's a codon on the mRNA, AUG, and this is the start codon. So in comes the ribosome. It comes in and binds to the mRNA right over the start codon. So the codon in yellow is what's being exposed, if you will. And the tRNA that's coming in right here is the tRNA that carries the complementary anticodon. Since there's an A in the codon, there has to be a U in the anticodon. A U in the codon, an A in the anticodon. A G in the codon, a C in the anticodon. This is the only tRNA that can come in and bind here. So it's the only tRNA that can donate an amino acid to the ribosome right here. And that amino acid is methionine. Now, they're showing us these three sort of oval areas on top of the large subunit, and these represent actual hollowed out areas inside the large subunit of the ribosome. So you have to imagine the large subunit in three dimensions, sort of like a large ball. And it's going to come in and it's going to land on the mRNA, and it's going to sort of cover the mRNA the way a ball would come in and sit on top of a piece of string. There are actually three hollow chambers inside that large subunit, and you can see that the tRNA has come into the middle chamber, or the one that's labeled P. Notice that the second codon, the one that they're showing us in black, sits right underneath this chamber, this hollow chamber. And another tRNA is coming in. For the second codon, UUA, we need an anticodon that reads AAU, and that's what this tRNA has. So it's going to come in and land here and bring in its amino acid, which is leucine. We refer to this chamber as the A chamber, and this one gets called the P chamber because P stands for peptide. The peptide bond is the kind of covalent bond that the ribosome is going to build between the methionine amino acid and this incoming leucine amino acid. A peptide bond will get built between those two amino acids inside the ribosome. And that's what they're showing us here. The methionine will be joined with the leucine as these two tRNAs sit in these two chambers, these two sites, the A site and the P site. Now the ribosome is going to move. It's going to shift over one position. Here's the AUG, here's the start codon. Here's that second codon we saw a minute ago. And here's the next codon, GGU. So now what was in P has shifted over into this chamber, and we call this one E for exit. What was in the A chamber a minute ago is now in the P chamber. And a new tRNA is going to come in, and here it is. So the new codon is GGU, and that matches an anticodon of CCA, and that tRNA is bringing in a glycine. Whatever's in the P site will be joined to whatever's in the A site. So this glycine will be added on to this growing chain of amino acids. It'll be attached to the leucine with a peptide bond. Meanwhile, over here, this empty tRNA, the one that already donated its amino acid, it will exit, it will leave, and the ribosome will shift again. So here's the peptide bond being built between the growing strand here and the new amino acid, the glycine. The tRNA that has already donated its amino acid, it's going to leave the exit site, the E site, and the ribosome is going to shift again. So now we can see our growing polypeptide, our growing chain of amino acids, has methionine, then leucine, then glycine, then phenylalanine. What's coming into the A site now has a methionine on it because we've hit another AUG. Remember, AUG is the start codon, but it's also a code for an amino acid. It's for a methionine. So there can be more than one AUG in here. Whatever was in the A site shifts over to the P site, and then that shifts over to the E site, and then that leaves. And the ribosome shifts again and again, and we keep building this long chain of amino acids. And that keeps going until the ribosome hits a stop codon. At the stop codon, there will be a tRNA that will come into this A chamber. But it will be unlike any other tRNA in that it doesn't carry an amino acid with it. So there will be a tRNA that comes in that has an anticodon to match this stop codon. It will come in and it will bind here, but it has no amino acid on it. So when the ribosome attempts to make a peptide bond between the last amino acid, this arginine, and what's in the A site, there will be no amino acid to add. And at that point, the chain of amino acids will be released. The ribosome will disassemble, it will let go of the mRNA, and translation will be completed. And that's what you're seeing over here. The mRNA is just released from the ribosome. The ribosome will go and reassemble at the start codon of another mRNA somewhere. The tRNAs that were used in this process, they will all get loaded up with their appropriate amino acid again, so that they can participate in translation over and over again. And the new protein, this new chain of amino acids, will be processed, and it will be folded, and it will be assembled into its three-dimensional shape that will determine what job it will do as it's set off into the cell.